What if everything we thought we knew about how bodies form was incomplete? In a groundbreaking discovery that challenges the foundations of modern biology, researchers at Tufts University have uncovered a second code that controls biological form, not written in our genes, but in patterns of electricity flowing between our cells. Today, we'll explore how this revolutionary finding is transforming our understanding of regeneration, biological identity, and potentially unlocking new frontiers in medicine. For nearly 70 years, since Watson and Crick discovered the structure of DNA in 1953, we've believed that our genes contain the complete blueprint for building and maintaining our bodies. This central dogma positioned DNA as the master controller of biological form, the comprehensive instruction manual from which all bodily structures arise. But scientists have long been puzzled by certain biological phenomena that don't fit neatly into this gene-centric view. The most striking examples come from creatures with extraordinary regenerative abilities, like the planarian flatworm. These remarkable organisms can regenerate their entire bodies from tiny fragments, raising profound questions about how exactly they know what to rebuild and how to organize it. This mystery has now led to a fundamental reconsideration of biological development that goes far beyond DNA. Planarians are small, freshwater flatworms that have fascinated biologists for generations. Their regenerative abilities are nothing short of miraculous. If you cut a planarian into dozens of pieces, each tiny fragment knows exactly how to rebuild itself into a complete, perfectly proportioned worm. Until recently, scientists assumed this regenerative blueprint was encoded entirely in DNA and expressed through genetic pathways. But Dr. Michael Levin and his team at Tufts University's Allen Discovery Center have demonstrated something extraordinary. These flatworms maintain complex patterns of bioelectric signaling, essentially voltage differences across cell membranes, that serve as a separate, non-genetic memory system dictating body structure. These bioelectric patterns function like a distributed information network, guiding cells during regeneration and instructing them not only what to become but also where to position themselves within the larger anatomical context. What Dr. Levin describes as essentially a second code represents a fundamental shift in our understanding of biological development. While DNA provides instructions for building proteins, this bioelectric network stores and processes information about large-scale anatomical structure, where heads and tails should form, and how organs should be arranged. This discovery isn't entirely without precedent. As early as the 1940s, embryologists had observed bioelectric currents during limb regeneration in salamanders, but the technology to systematically investigate these phenomena remained limited until recently. The Levin Lab has pioneered new techniques for visualizing and manipulating these bioelectric patterns, revealing their fundamental importance in orchestrating complex morphological outcomes. These patterns don't replace genetic information. Rather, they work alongside it, providing a different kind of information that genes alone cannot encode: spatial relationships and three-dimensional patterning that guide development and regeneration. The research team used a combination of voltage-sensitive dyes and pharmacological agents to visualize and manipulate the bioelectric networks in planarians. By temporarily disrupting specific ion channels without altering the worm's genetic makeup, they created something remarkable: planarians with two heads, one at each end. But the truly revolutionary finding came next. When these two-headed worms were cut into pieces, the fragments regenerated into more two-headed worms, even though their DNA remained completely unchanged and the pharmacological disruption had long since cleared from their systems. This demonstrates unequivocally that pattern memory can be stored in a medium other than DNA. The bioelectric network had effectively been reprogrammed, with new instructions that persisted across generations of cells. The persistence of this altered bioelectric pattern represents a form of non-genetic inheritance that challenges conventional understandings of how biological information is transmitted. The two-headed state became self-perpetuating, with the altered bioelectric pattern instructing new cells to maintain the same abnormal configuration. Further experiments revealed that these bioelectric patterns operate through a distributed network of gap junctions, specialized channels that allow direct electrical communication between adjacent cells. When these gap junctions were temporarily blocked, the worms lost their ability to maintain coherent bioelectric patterns, resulting in severe defects in regeneration. This finding suggests that collective cellular communication, rather than individual cellular decision-making, governs the formation of large-scale patterns. It's not individual cells making isolated decisions about what to become. Rather, It's the coordinated communication across networks of cells that allows complex patterns to merge and be maintained. This collective communication system enables cells to understand their position within the larger whole, something that cannot be achieved through genetic information alone, which is identical in every cell of the organism. This discovery bridges multiple fields, drawing unexpected connections between developmental biology, neuroscience, information theory, and even artificial intelligence. The bioelectric networks function remarkably similarly to primitive neural networks, processing pattern information in much the same way brains process sensory data. This parallel suggests evolutionary connections between the mechanisms that maintain bodily form and those that process cognitive information. From an information theory perspective, the findings demonstrate that living systems utilize multiple, parallel systems of information storage and processing beyond the genome. While DNA provides the instructions for building the components of the bioelectric network, the ion channels, gap junctions, and pumps, the network itself stores and processes a different kind of information, spatial relationships and pattern configurations that cannot be directly encoded in linear genetic sequences. This multi-layered information processing suggests that biological systems are far more complex computational entities than previously recognized. For regenerative medicine, these insights open entirely new avenues for therapeutic intervention. Rather than focusing exclusively on genetic or biochemical manipulations, future approaches might target bioelectric patterns directly to guide tissue regeneration. Early experiments in the Levin lab have already shown promising results in frogs, where manipulating bioelectric signals induced limb regeneration capabilities not ordinarily present in these animals. This suggests similar networks may exist across many species, including humans. As Dr. Levin explains, we're not just talking about healing wounds faster. We're exploring the possibility of reprogramming the body's own bioelectric patterns to achieve what was previously thought impossible: regeneration of complex structures in mammals. Current approaches to tissue engineering typically involve providing cells with scaffolds and growth factors, essentially attempting to micromanage the rebuilding process. The bioelectric approach suggests a more elegant alternative. Rather than dictating each step of regeneration, future therapies might restore the appropriate bioelectric pattern, allowing the body's own intelligence to orchestrate the complex process of rebuilding damaged tissues and organs. Perhaps most intriguingly, the computational properties of these bioelectric networks suggest new architectures for artificial intelligence systems. Unlike conventional computer algorithms that process information sequentially, bioelectric networks operate through distributed, parallel processing, more akin to neural networks but with unique properties that might inspire novel computational approaches. The way these networks store and process pattern information, maintaining complex three-dimensional structures through distributed electrical signaling, could potentially inform new approaches to machine learning and artificial intelligence. These biological systems have evolved sophisticated information processing capabilities over millions of years, potentially offering insights that could help overcome current limitations in artificial intelligence design. The parallel, distributed nature of bioelectric computation offers a glimpse at information processing systems that are both robust and adaptable in ways that current AI systems struggle to achieve. The discovery raises profound philosophical questions about biological identity and the nature of the self. If a significant aspect of what makes you exists in bioelectric patterns rather than solely in your genome, it suggests a more fluid and dynamic understanding of biological identity than previously recognized. As Dr. Catherine Pittman, a philosopher of biology at Oxford University, notes, we're accustomed to thinking of ourselves as the product of our genes. But this work suggests that what we call self is maintained through multiple interacting systems of information storage and processing, only one of which is genetic. This multi-layered view of biological identity challenges reductionist approaches that seek to explain organisms solely in terms of their molecular components. Instead, it suggests that emergent properties arising from complex interactions between different information systems, genetic, epigenetic, bioelectric, and possibly others yet to be discovered, collectively define biological form and function. The philosophical implications extend to questions of biological individuality and the boundaries of organisms. If bioelectric patterns can be manipulated to create sustainable alterations in form, where does one individual's form end and another's begin? The ability to induce two-headed planarians that stably reproduce their altered morphology challenges conventional notions of species-typical form and raises questions about the fluidity of biological identity. As this research field expands, It promises to redraw the boundaries between developmental biology, neuroscience, and information theory, offering a new lens through which to understand the ancient mystery of how complex form emerges from seemingly formless cells and how it might be restored when lost. The bioelectric dimension of life, long overlooked in favor of molecular and genetic explanations, may hold the key to unlocking regenerative capacities that have remained dormant in humans since our evolutionary past. Future research will likely focus on mapping the bioelectric codes of more complex organisms, including mammals, and developing more sophisticated tools for visualizing and manipulating bioelectric patterns in living tissues. The field also faces important challenges, including developing standardized methods for measuring and interpreting bioelectric signals across different cell types and organisms. Despite these challenges, the discovery of bioelectric coding as a fundamental aspect of biological development represents one of the most significant paradigm shifts in modern biology, one that may ultimately transform our understanding of life itself and open new frontiers in medicine, artificial intelligence, and our philosophical conception of biological identity. The discovery of bioelectric signaling as a fundamental code governing biological form represents nothing short of a revolution in our understanding of life itself. Beyond the genome lies this second, equally crucial system of information, patterns of electricity that guide cells in their collective effort to build and maintain complex bodies. This insight transforms multiple fields simultaneously. Developmental biology now has a new dimension to explore. Regenerative medicine gains powerful new approaches to healing. Artificial intelligence finds inspiration in nature's distributed computing networks, and philosophy must reconsider what constitutes biological identity and selfhood. As we continue to decode these bioelectric patterns, we may unlock regenerative abilities that have remained dormant in humans, potentially revolutionizing medicine. The implications extend far beyond science, challenging us to reconsider the very nature of what makes us who we are. Our bodies, it seems, are not simply the product of our genes, but emerge from the complex interplay between multiple systems of information, a dance of molecules and electricity that maintains the pattern we recognize as ourselves. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.